here, we began speaking about that third phase of glucose oxidation. It's called the electron transport chain. And this requires oxygen. So this is oxidative phosphorylation. We said that about 4 net ATP were gained in the earlier stages. Okay, got it. So someone wants a little bit of guidance in terms of how the exams will work, and I'll do that towards the end. So look out for that. Thank you. So we said the earlier stages of glucose oxidation, glycolysis, the Krebs cycle did not yield very much ATP, about 4 net ATP if we consider what we have to invest. So the bulk of the ATP is actually formed in this third phase that we're going to talk about. This is the electron transport chain, and we kind of briefly described this when we left off. We said that this was where the electrons were passed from complex to complex. These complexes were embedded in the inner mitochondrial membrane. And when they're passing the electrons from one to the other, this generates some energy to move hydrogen ions, creating a gradient, which will then favor the synthesis of ATP. Now, this yields much more ATP. This is a much more efficient process. But the caveat here is that it requires oxygen. The earlier stages, glycolysis and the Krebs cycle, did not require oxygen. That was substrate level phosphorylation. And if you remember our 2 scenarios, our 2 options for creating ATP, one was substrate level, and that was where we borrowed or stole, we took an electron from another carrier molecule. The exact name of that molecule is not important, but as we move on, I'll talk about some examples about what those carrier molecules are. And we could compare that to oxidative phosphorylation, which is where we find a free-floating inorganic phosphate, inorganic in that it's not attached to another organic molecule, and we use that to phosphorylate ADP and make ATP. So that is the mechanism by which we make ATP here in oxidative phosphorylation. So again, just to talk about the electron transport chain, I think we also summarized this when we left off. So we're actually going to get into the details of this and see what's actually going on. Okay, so here we are looking at the electron transport chain. Okay, and we're really looking at what's happening across the mitochondrial membrane. So kind of for some context here, this is our mitochondria, right? It's this ovoid shaped organelle within the cell, sometimes referred to as the powerhouse of the cell. We'll explain why that is as we go through today's discussion. It's got a double membrane, so although it has this outer membrane, it also has a highly folded inner membrane. Like so. And so we're looking at what happens across these 2 membranes, the outer mitochondrial membrane and the inner mitochondrial membrane. So this is the area that we're really focusing in on, these 2 membranes, the outer and the inner, okay? So let's kind of zoom in and draw this out a little bigger, these 2 membranes. So here's our outer mitochondrial membrane. And then here's our inner mitochondrial membrane. Okay. So the first thing, so let's label these off first. So this is the outer mitochondrial membrane. And this is the inner mitochondrial membrane. Okay, so the first thing we want to establish is that we have the presence of hydrogen ions in higher concentration in the intermembrane space. So this space in between both of these membranes, we refer to this as the intermembrane space. And in this space, we have a high concentration of hydrogen ions or protons. Okay, we'll talk about why we end up with a higher concentration here in a bit, but we've got a higher concentration of hydrogen ions in the intermembrane space. Okay, and we've got a lower concentration of hydrogen ions down here, which is actually in the mitochondrial matrix. Right, so not very many protons here. We'll talk about how we end up with this gradient in a bit. So this area out here is called the mitochondrial matrix. Okay. Now, the actual electron transport chain, and we're going to really think about that word or that term, those words, is really the passing of electrons across these complexes. So we have a series of complexes that are embedded in the inner mitochondrial membrane. You don't have to really know the details of these complexes. There's complex 1, there's node 2, and then there's 3. And so on. I'm not going to ask you to know the details of the complexes, but certainly the overall picture of what's actually happening. And then the final complex is the most important one. This is called ATP synthase. And this is kind of where all the magic happens. Okay, so the final complex is the one that I want you to kind of be focused on in terms of knowing the name. This is called ATP synthase. Okay, and this is the most important complex. Now what happens is, if you remember our discussion, we talked about the electrons being shuttled from glycolysis. So remember, glycolysis created electrons by reducing our NAD plus to NADH carrying electrons. We also had electrons generated in the linking step as well as in the Krebs cycle. So all of those electrons that are being shuttled by our carrier molecules, NAD, and in the case of the Krebs cycle, we also had FAD, make their way into the mitochondrial matrix, and this is kind of what they're doing. So we have those electrons that make their way into the matrix. So we have NADH. Remember, this has electrons that are being shuttled, as well as FADH2. When we oxidize these electrons, we oxidize these carriers, we can then free up the electrons. So the electrons that get freed up, they're passed to the first complex, and then on the other side, we end up with the oxidized form of these molecules again. So we pretty much just grab the electrons off of these carriers, return them back to their oxidized form, and then the electrons make their way from one complex to the other, okay? And this E is standing for those electrons that were taken from these carrier molecules. So we pass them to complex 1, they're then passed to complex 2, and so on and so on. Now, the gradient that we spoke about, so the hydrogen gradient, the fact that we have more hydrogen ions in the intermembrane space than we have in the mitochondrial matrix, is created because of the movement of these electrons. okay? So as we move electrons from complex 1 to complex 2, we're simultaneously pumping protons from the mitochondrial matrix into the intermembrane space, okay? So we've got these hydrogen ions that are being pumped each time we move electrons from the mitochondrial matrix up into the intermembrane space. And that is how we end up with this hydrogen ion gradient, where we have more hydrogen ions in the intermembrane space and less hydrogen ions down here in the matrix. Okay, now this gradient is going to be very important to the actual synthesis of ATP. Because we now create this hydrogen ion gradient, all of the protons now really want to get back to this side. They really want to get back into the matrix because of this strong gradient that we've created. And so when they do that, they go down ATP synthase, these hydrogen ions, because of the strong gradient. And that happens to create the ATP from ADP. So the energy of all of these protons moving down ATP synthase is what actually phosphorylates ADP to create ATP, okay? Now, in order to get the electrons, in order to get the movement from the final complex, we have to have oxygen present here. This is where oxygen comes in. So oxygen is just standing by here. Here's our molecule of oxygen. And oxygen is going to accept the last electron. It's going to be the final electron acceptor. And so the electrons are going to go from the final complex on to oxygen. This is what keeps the chain moving. If there's no oxygen, then the electrons kind of pause at that final complex, and there's no one standing by to grab those electrons to keep this chain moving so that we can continue to synthesize ATP. This is the whole purpose of breathing in oxygen. It has to act as the final electron acceptor in order to keep the electron transport chain moving so that we can continue to synthesize ATP. And we know that ATP is crucial for cellular work, okay? So the movement of these hydrogen ions down ATP synthase is what phosphorylates ADP. Remember, we have ADP plus the inorganic phosphate, and that can then be phosphorylated to create ATP, okay? And so this is the whole purpose of this entire chain, the making of ATP via ATP synthase. All right, so I'm gonna pause here and see what initial questions come up. And then I'll kind of recap this again just to add some more clarity as we go. Okay, I don't see any questions, so I'm going to keep going here. So again, just to recap what's happening here, we have the mitochondria, and this is why the mitochondria is called the powerhouse of the cell. This is where the energy is actually synthesized, at least most of the energy. This process synthesizes anywhere from 28 to 32 ATP, okay? We'll talk about why there's a discrepancy in that value, but anywhere from 28 to 32 ATP is synthesized here via the oxidative phosphorylation. And this is why we call this process oxidative phosphorylation, because there must be oxygen here so that this chain can keep moving and we can create this hydrogen gradient. Okay, it's kind of like a domino effect happening here. So the movement of electrons is what creates the gradient. And then the gradient is what actually activates ATP synthase and phosphorylates ADP in order to make ATP. And if we don't have oxygen, we don't have electrons moving. And if we don't have electrons moving, we don't have a gradient, Okay, we don't have this hydrogen gradient.